Thank you. It's indeed a great honor to have been asked to speak in the series. I looked at some of the earlier speakers and the topics, and I hope I can do justice to the series. I was trained as an electrical engineer, and electrical engineering, as many of you know, derives its ancestry from physics, and hence EEs have inherited the ability of physicists to poach in the sciences, in technologies, and even in the social sciences. So my confession is that for the last 25 years or so as an academic researcher, I have been an incessant poacher. And so when I was asked to speak in this series, I considered three possible topics. One was these historical pieces on linear programming that I have already written on. And another topic that I thought of was this book with John Hooker, which actually we had a subtitle for the book which the publishers didn't allow. The subtitle was Eating Sauerkraut with Chopsticks. Because the idea was, optimization came from the right brain, kind of spatial reasoning and geometry, whereas logic really is sort of a left brain, very symbolic kind of field. And then the third topic that I thought I could talk about was these crossings in computation and biology. And I chose the third, partly because I haven't had an opportunity to write or to think about this in an integrative fashion till now, and I thought this would be an opportunity. But that also necessarily means that this lecture is a bit exploratory and perhaps a little preliminary, and I hope you'll forgive me. I see a lot of very distinguished biologists in the audience, and I apologize in advance for some of the elementary issues that I'm going to raise here. My personal exploration of computational biology began in the mid-90s when my colleague, Professor Preeti Shankar at the Institute, and I arranged a lecture series leading to a JNC discussion meeting on the subject in December of 1995. This theme of computational biology also reflects my work at Strand Genomics, the company that we spun out over the last 3 years, where we have built an interdisciplinary team of really outstanding computer scientists and young biologists. And it's been fascinating that we've had to sit around in these interdisciplinary teams with physicists, chemists, biologists, and computer scientists, and get past the jargon, the cultural and other barriers, and learn to work together. Freeman Dyson, in his New York Public Library lectures, entitled The Three Faces of Science, later published by the Oxford University Press in 1999 as The Sun, the Genome, and the Internet, says that today the most successful craft industries are concerned with software and biotechnology, and which certainly struck a chord when I read that. And Donald Knuth, the outstanding and multifaceted computer scientist from Stanford, at the ACM Vision for the 21st Century conference in Boston in 1999, stated that biology should keep computer scientists busy for at least 50 years. And finally, at a recent meeting, a very recent meeting actually at TJ Watson in Yorktown Heights, I learned that one prevalent view within IBM is that in 50 years, people will assume that computers and computing were actually developed for biology. So, in some ways, these strands were the motivation for this lecture. What I will do is give you a slightly biased history of computer science, a slightly biased history of modern biology, and then talk about some of these crossings that I've witnessed and that I hope will lead to interesting developments. So on the biased history of computer science, and this bias is really primarily from my training and interest in logic. Computer science really goes back to Leibniz, and who formulated two very interesting ideas. He actually talked about something called the universal characteristic and the calculus ratio conator. So the universal characteristic was supposed to be this language in which you could express all problems, and the calculus ratio conator is the engine by which you actually compute and deduce all truths from which are stated in this universal language. And this, I believe, was the foundation of computer science. And he actually went on to try to come up with both constructs. Of course, these were very early works and didn't actually serve the purpose that they were intended for, but then started the work. Later, the work of George Boole really converted logic into algebra, right? And Frege really gave the formal syntax for logic to be formulated. The for all and there exists, and then the ways of writing predicate formulas which were introduced by Frege. David Hilbert, and now we're at the turn of the 20th century. There is a problem, seek its solution, you can find it by pure reason. Okay. So this, his famous line was, of course, in German, wir müssen wissen, wir werden wissen. So we must know, we shall know. And in fact, this is inscribed on his tombstone. So, of course, Hilbert's dreams were dashed by Gödel, and Kurt Gödel, who came up with the theories of incompleteness and undecidability. It's very interesting that apparently, of course, when Gödel and Einstein used to walk together in Princeton, Gödel would look very proper, well attired and carrying a leather briefcase and so on. So people would refer to them as Einstein and his lawyer, right? And Einstein once said that Gödel has gone completely crazy. He voted for Eisenhower. So, but Gödel, with great precision, proved this amazing theorem. And in the process of proving this theorem, actually, because it's a mathematical result, he needed to show that the problem he was addressing could be stated as in the same language. And therefore, he actually, instead of, he knew that it would be difficult to convince people of his result. And so he wrote it down in an extremely meticulous and careful manner in the style of a programming language. So actually, in some sense, Gödel was the first person to actually write down a formal programming language, a high-level programming language. But of course, the great breakthrough for computer science came after Gödel, and that was Alan Turing. And Alan Turing learned about Hilbert's Entscheidungs problem and Gödel's theorem in a course given by Newman in 1935 at Cambridge. And it was apparent, if you saw both statements in the same course, of course, that inherently Gödel's theorem must disprove Hilbert's thesis. And but in order to prove it, Turing needed a construct. And the construct he came up with is now known as the Turing machine. And he constructed the Turing machine model and proved Hilbert's dream was impossible. In fact, Turing showed something much more powerful. What he showed was this notion of a universal machine. And here's a quote from one of his papers. Let us now return to the analogy of theoretical computing machines. It can be shown that a single special machine of that type can be made to do the work of all. It could, in fact, be made to work as a model of any other machine. The special machine may be called the universal machine. And this is a very important idea, because what sits on your desk today as a computer is a universal machine. And the fact that it does so many things and not just calculates your bills is the fact that it derives from this conception of a universal machine. Now, of course, the person who really understood the import of Turing's result was von Neumann. And von Neumann actually was called in to review the ENIAC project, where a computer had been built at the University of Pennsylvania. In fact, I taught in that building for a year in Moore Hall, as it's called. And the model of the ENIAC still sits there. But when von Neumann came and reviewed the ENIAC project, he actually developed a little paper for the next generation, which was called the EDVAC project. And in that little note, he described in a very careful, logical manner how one could build a universal machine, and that has come to be known as a von Neumann architecture. And this was way back in 1946 or 47, and Alan Turing, of course, had done his work on the universal machine also earlier in the 40s. But in spite of all of these observations and insights, most people didn't quite understand the power. And someone as distinguished as Howard Aiken, who headed, of course, the Harvard Computation Lab, made a fairly preposterous statement in as late as 1956. He said, if it should turn out that the basic logics of a machine designed for the numerical solution of differential equations coincide with the logics of a machine intended to make bills for a department store, I would regard this as the most amazing coincidence I have ever encountered. And of course, he had missed the point altogether of the universal Turing machine. So finally, who actually invented the computer? Well, in many accounts you will read about, I guess, the Joseph Marie Jacquard, the Jacquard loom, which was a programmable loom, as being the first sort of computational engine, and then Babbage and Lovelace and Shannon, Aiken, lots of contributions up to Eckert and Mauchly, who actually built the ENIAC, and then Goldstein and von Neumann, who actually critiqued the ENIAC, and this led to really the modern building of the modern computer, which were initially known as Johnniacs because of von Neumann's involvement. Turing himself actually designed a machine called the ACE, and in fact was very critical of von Neumann's work, and said that the American tradition of solving one's difficulties by means of much equipment rather than thought was what he had to say about Johnniacs. But really, sort of the Johnniac was really a blending of these principles of very clear logical thought and engineering. I mean, whereas I think if Turing had had his way, what you'd have had is a very elegant but spare machine. In fact, many of the ideas in his design of ACE have found their way into what are called reduced instruction set computing. So the RISC machine is really very similar to the ACE concept. Well, so this was how computers came to be. But what then have been some of the interesting, more modern constructs in computing? To me, the work that happened in the early 70s, 1970s, which is a little more technical to describe, the so-called NP completeness framework, which is actually postulated by Cobham in England, Edmonds in Canada. Well, actually, Edmonds was in the US at that time, at the National Bureau of Standards. Stephen Cook at Berkeley and Leonard Levin in the Soviet Union, in Russia. It's a beautiful theoretical framework that paralleled the original contributions of Gödel and Turing, but in the context of practical computation. So whereas Turing and Gödel were talking about what is computable at all and what isn't, this NP completeness theory was trying to mark out the boundaries of what was practically computable. Okay. And this theory of NP completeness has led to people looking for more powerful models of computation, and I believe that the two most exciting ideas today are those of quantum computation and DNA computation. So quantum computing was formulated by Feynman, actually, in 1982, who had, of course, a lot of interaction with Fredkin at MIT at the time. And I was a PhD student just finishing up when Feynman gave the famous lecture on quantum computing. He called it qualitative probabilities, and that was in the title of the lecture. DNA computation was something that, again, was developed by a computer scientist, Edelman, who is the A of the RSA algorithm, which is the famous encryption method. Edelman at some point had left MIT and gone to the University of Southern California, where he became an active researcher in biology. And actually, initially, I think he was motivated to work on HIV, and then later, developed this technique of DNA computation. And I'll say a little more about that when I get into the detailed topics. Basically, the ideas of Edelman have led to work by Eric Winfrey at Caltech, which in 1998 actually showed that using a molecular tiling method, a kind of self-assembly of molecular tiles, one can manage to simulate a universal Turing machine. So again, the idea has come back to the universal Turing machine. So now let me switch to biology and do a very similar quick run through, again with certainly my own biases here. And since I claim to be talking about modern biology, I can safely skip, I guess, natural selection and theory of genetics. 1953, the year I was born, I guess, was the big year when the structure of the DNA was worked out. I guess this was, and of course, this is the 50th anniversary, so there are many celebrations going on. There was also, of course, very important work on the structure of proteins in the 1960s, which has now become, great challenges in high throughput technologies for working out protein structures, and also structure prediction the so-called protein folding problem, and with contests at Asilomar that happen every 2 years, and that's another very vibrant area. And again, I'll talk a little bit about the protein folding problem later. But I thought the topic that for a general audience, the topic of most interest in modern biology is clearly the Human Genome Project, and so let me say a little bit about at least what I know of the history of that. In fact, in the mid-1980s, Charles DeLisi, who headed the life sciences program at the Department of Energy, initiated the Human Genome Project. This is not a fact that's very well known that it actually began at the Department of Energy, and because they couldn't convince NIH to back this project. And so Charlie Cantor, who actually happens to be on our scientific advisory board at Strand, talked Charles DeLisi, who also happens to be on our scientific advisory board, to actually fund this program. In 1988, Watson gives testimony to US Congress and promises to spend at least some of the money on ethics and issues related to ethics. So the Congress gives him the go-ahead, and NIH jumps into the game, and NCHGR gets set up, and so now it becomes primarily an NIH initiative. In the early 90s, the French, Cohen and Weizenbach, complete a first mapping to be refined later to a high-resolution map, reflecting the approach of map first and then sequence later. So the first real sort of breakthrough comes from France. In 1992, Watson resigns over patenting issues, and Francis Collins takes over, as Craig Venter leaves NIH to form Tiger. In 1996, the Bermuda Principles are formulated. This is a group working on the public Human Genome Project, coming to a decision that genome data will be public. Very important landmark meeting. In 1998, a gauntlet is thrown by Venter and Eugene Myers at Celera. They claim to have a new technique, the whole genome shotgun method, which they claim will race ahead of the public project. And everyone gets very tense because there's a private company. If they get there first and then they patent all these genes, then it really stifles research. So finally it becomes a very political decision, and the decision is made that the patenting will not be allowed. It will have to be a public information. So, but in any case, the gauntlet thrown by Venter actually leads to the public project responding, with great energy. The Whitehead Institute, Genome Institute at MIT, the Sanger Center near Cambridge, Baylor College of Medicine, Washington University in St. Louis, and the DOE's Joint Genome Institute, all band together and go after the human genome, the public project. And Chromosome 22 is published. All this in 1999, and in 2000, the joint announcement finally, Venter and Collins, of the completion of the draft, and finally, I guess Ari Patrinos at the Department of Energy had to, reconcile these two people and make them come together to make the joint announcement, because it was war, right up to that point. But in any case, the remarkable thing that has happened, of course, is that now the genome is published, and I think in Nature on 15th of February, and by the public in the Human Genome Group, and Celera published it on 16th of February in Science. So they both came out at the same time. The reactions to the publication of the human genome sequence, Bill Clinton's reaction, right? He says, it is conceivable that our children's children will only know cancer as a constellation of stars, right? Very poetically put. And the Hindustan Times says, however, many are concerned about the resulting risk of discrimination in insurance and employment, leading to nightmare dilemmas about whether such tests should be permitted by employers, lenders, and health insurers. So, there's a lot of excitement, but there's also some fears about where this will all lead. I think there are some interesting books now that are actually coming out. There's a recent one by Francis Fukuyama, and I'm afraid I'm forgetting the title, that actually talks about some of the implications, and particularly the ethical ones. One of the great things, of course, about the Human Genome Project is that many techniques were developed as part of this project. The craft of biotechnology has really sort of blossomed. And if you think about sequencing technology, which was originally started, I guess, by almost simultaneously by Fred Sanger and Walter Gilbert in the 1970s, it took almost till 1986 till there was a real improvement in that technique, and that was the work by Leroy Hood at Caltech, who introduced the first sort of automated sequencing machine. In 1990s, though, because of the Human Genome Project and so on, you saw fantastic improvements in technology, and the high throughput capillary sequencing machines that were generated as a consequence. Also, the computer science that developed around it, the algorithms for assembling fragments, and for the shotgun technique, which came from Gene Myers, were also, highly honed and very efficient. It also became clear that there were several operations that could be performed with ease on DNA, and synthesis of desired strands, separation of strands by length, merging of extraction, melting, annealing, amplification. So amplification is, of course, the use of PCR to make copies, cutting with restriction enzymes, ligation, and detection. And the facility with which people could play with these operations has also something to do with the fact that things like DNA computation are now possible, because it is these primitive operations and techniques that are used to construct DNA computers. Now, the Human Genome Project and the publishing of not just the human genome, but so many, I think somewhere on the order of 30 to 40 genomes now have been fully sequenced and published, of microbials, of Drosophila, of C. elegans. There are a whole bunch of models now that one can play with at using the genome information that is publicly available. Now, this is leading to a very rapid development of fields like functional genomics, comparative genomics, and we will actually see the benefits of these coming at a highly accelerated pace now. If you actually go into a place like Whitehead today, it's just banks and banks of computing, and enormous amount of effort going into computing, and Eric Lander sits, almost half the time at the lab for computer science because he has all these interactions now with the computer science department. The virtual cell projects and various systems biology projects have also started up, and this reflects more and more the comfort or that the life sciences are starting to have with computational techniques, and I'll get back to this later, but I think these are some of the emerging areas where there's an interesting crossing between computing and biology. So let me very briefly touch on three topics in a little detail. I guess first crossing that I would like to talk about is the whole issue of how information is contained in sequences, right? Which is basically something that computer scientists have always been concerned with, and certainly people who work in genetics and genomics are also. In fact, let me use a very interesting anecdote going back to, I guess, Sanger. In 1977, Sanger and his co-workers at MRC's molecular biology lab in Cambridge accomplished the remarkable feat of decoding a complete biochemical message. The team's success was in deciphering the genetic text of one of the smallest bacterial viruses. I think it's called 0x174. Is that, I don't know how it's actually pronounced. A mystery had surrounded this virus. It did not seem to contain enough information for making the 9 different kinds of proteins which are in fact produced when the virus infects a healthy cell. The sequence of 5,375 chemical symbols on the DNA of the virus was too short to spell out the full set of instructions to the cell. The mystery was solved, but the encoding was particularly tricky as the words of text actually overlap so that more messages could be squeezed into a small space. So, the start codons, codon is of course ATG, and the stop codons are TAA, TGA, and TAG. In this virus, 2 distinct genes are coded just by shifting the reading frame by 1 letter, right? So actually, you're coding for 2 genes, but using the same amount of space, +1, right? And similarly, there are genes that overlap, maybe not so completely, but just at the ends. And so essentially, it's the encoding is almost like a tight crossword puzzle, right? Although it is one-dimensional. Actually, Freeman Dyson uses a very nice analogy. There's apparently a Mozart violin duet, which is very cleverly composed, so that you place 1 sheet of paper of music on a table, and the 2 violinists stand facing each other, and you read, one reads it top down, the other reads it bottom up, and it actually plays as a duet. And so he must have had a lot of fun actually composing that, but it also represents a way of putting information together. So Dyson actually refers to Sanger's virus as Sanger's Mozart virus, and but all of this actually leads to 2 fundamental questions, I think. One is it's related to linguistics, and the other to genetics. So the first question is, how is it that a child can acquire something as complex as language so quickly by being exposed to random samples of adult speech? Okay, that's a fundamental question. The second question is, how is it that something as complex as a human being can evolve in a relatively short period of time by random mutations of genes? Right? So Chomsky has answered the first question, though some might still debate his answer, that there is an innate grammar or production system that predisposes the child to form a correct theory of language, and this has come to be known as the language instinct. Right? People are searching for a biological basis of that. So the question is there then a production system that assists natural selection? Okay, is there a grammar that assists natural selection? Jeremy Campbell, in an old but very interesting book called Grammatical Man in 1982, actually states that grammar is an anti-chance device which allows plenty of room for novelty. Right? A mutation of a rule in the grammar could lead to an innovation, a burst of originality. So if you are allowing actually the grammar itself to mutate a little, right, that could actually have enormous effects. I would think that in order to settle debates like this and to look at questions like this, you would eventually have to use some techniques from computer science. And the second topic that I'd like to touch on is protein folding, the holy grail. Right? Proteins are the most important biological molecules in some sense because they are responsible for functioning in all living organisms. Each protein has well-defined functions which range from building up DNA and RNA molecules to controlling different parameters in living cells. It's amazing that proteins are built of very simple building blocks known as amino acids. There are 20 different amino acids, each of which consists of a central carbon atom called the alpha carbon, which is bonded to an amino group, a carboxyl group, and a side chain. The side chain is different in each amino acid and is solely responsible for the characteristics of the amino acids like shape, size, and polarity. The structure and properties of a protein in turn depend on the basic features of the constituent amino acids. These amino acids are linked to each other by means of peptide bonds, and they result in a constrained backbone of the alpha carbons, and they can twist with limited values for the bond angles. In fact, the late Professor Ramachandran determined what constraints there are on these bond angles, and these came up with what is come to be known as the Ramachandran plot, which basically look at conformations that do not violate steric conditions of minimum interatomic distances, so the van der Waals forces and so forth. Actually, these plots are akin to things that we look at in robotics. So you could think of this as a robot joint, and in robotics we call these configuration space diagrams, and they're really it seems like these two different fields have obviously, well, these are kinematic assemblies, so it's not surprising that some of the same techniques have sprung up in both fields. Proteins fold into compact and very varied structures, but these are usually combinations of simple motifs called secondary structures. The motifs are of two types, helices and beta sheets. And the final protein structure can be viewed as being built up of these secondary structures, connected in specific ways. Now, the determination of the structure of proteins is a very important problem, and the three-dimensional structure of the protein is believed to be, of course, a very important determinant of the properties of the protein. So finally, it all depends on how this protein goes and and binds with other proteins, and how various, for example, if you're ingesting drugs, then how these drugs will go and bind with these proteins, and the metabolites that will be created, and so forth. So if you are able to actually predict the structure of the protein just given the amino acid sequence, that could be a very, very powerful idea. And that's precisely what has come to be known as the protein folding problem. And the most remarkable discovery in this area was made by Affinson and his colleagues way back in the 1950s, when they found that many simple proteins had a unique native structure, which just seems to depend on the sequence. Right? And this has been subsequently verified for a large number of proteins, and is now believed to be the native structure. It's now believed that the native structure is actually the minimum energy configuration. Right? This is also known as the thermodynamic hypothesis. So this has led to an enormous interest in trying to develop methods to predict the three-dimensional structure, and this is the race for so-called protein folding algorithm. And determining a protein sequence is, of course, a fairly easy problem, but determining the three-dimensional structure is still very slow and very expensive. And I believe today the state of the art is still fairly far from coming up with perfect structures, but there is a contest held every 2 years. It's called CASP, and people are now able to predict maybe 70 to 80% of the structure correctly, but it's still very far from being useful in a very general way. The IBM, in fact, actually has invested a great deal of money in what is known as the Blue Gene project. And in fact, it is probably not very well known that they actually had the famous parallel computer that played chess, right? The Deep Blue that actually beat who was that? Right, Casper. And the reaction to that, the public reaction to that, was enormous. I mean, actually people got really frightened about this whole idea that machines will take over the earth, and so IBM pretty much shelved that project because they just felt that it would have a lot of negative image for the company, and so suddenly the protein folding problem appeared as the panacea, right? So what they've done is actually moved that entire group that worked on Deep Blue now to Blue Gene, and so now they're all working on trying to solve the protein folding problem. So it's actually become the problem on which high performance computing is being benchmarked. Right? So IBM will actually build more and more powerful computers, basically, to attempt to solve this problem. But in the process, they'll also be developing their technology. Now, I just wanted to mention that this connection between computer science and the protein folding problem, well, one connection is, of course, this fact that protein folding is now defining how high performance computing evolves, but also there is this notion of the Leventhal Paradox, which basically relates to the fact that that there are a large number of choices in how a protein could fold, and the fact that in spite of although there are these exponentially large number of choices, proteins do fold is sometimes referred to as the Leventhal Paradox. But or more precisely, I guess, the view that an intermediate conformation evolves via a sequence of somewhat random modifications leads to a paradox called the Leventhal Paradox. And Martin Karplus and his researchers, and then a bunch of others, have actually tried to explain this paradox now using this whole theory of NP completeness. And they've actually shown that various models of energy models for proteins, if you try to look at the problem of minimizing that energy function, that problem turns out to be an NP hard problem or an NP complete problem. So thereby explaining, at least in some sense, the reason why we have not been able to, come up with a good solution for protein folding on digital machines, but also deepening the mystery in some sense, because now the question is, what is going on that allows proteins to fold? So it's very likely that a better understanding of the nature of dominant forces would actually lead to better success in predicting the three-dimensional structure. So there is an interplay going on now between computer science and molecular biophysics, but the mystery is still unresolved. I think I've already mentioned something about DNA computing. Let me just say a couple of things and then try to close up here. In order to determine whether massively parallel DNA computations might prove useful for the solution of hard problems like NP complete problems, Edelman encoded and evaluated a particular type of hard problem known as the Hamiltonian path problem using a generative algorithm. Basically, he showed how to encode a graph. In this case, this was a small example of graph with about 7 nodes in the graph, and connected by about 6 edges. And he used one set of oligonucleotides that represented the nodes and a partially overlapping complementary set of oligonucleotides that represented the edges connecting these nodes. And these oligos were allowed to hybridize in solution and ligated together into paths. And the paths were amplified using, of course, PCR kind of techniques, and then successively sieved again by hybridization to determine whether the paths that contained all the nodes at least once were present. These are what are called Hamiltonian paths. The presence of an oligonucleotide that met these demands correctly revealed that a Hamiltonian path through the graph did exist, and that's the problem. We are trying to determine whether such a path exists or not. This was done in 1994, and Edelman actually walks around with this little kit bag with test tubes and so on, and shows how you can actually build a DNA computer. And it's of course led to a lot of work, and people like Richard Lipton at Princeton and others have developed this technique much further, solving problems like satisfiability problems in logic and so forth. I believe the largest problem that has been solved is that recently a 20 variable instance of 3-SAT, which is a particular variation of satisfiability problem, was solved on a DNA computer by Edelman's group. And of course, if you were to actually enumerate all solutions for this problem, there would be more than 1 million possibilities. So this actually searches through them very efficiently. So I hope I've given you some sense of why these two fields, in some sense, have been developing on their own and have had their own internal dynamics, of course, but then they are crossing every now and then, and there are some very interesting ideas and techniques that come out of these crossings. I believe that some of the most interesting future crossings will be probably in this whole idea of emergent properties of complex systems, or what has come to be known as systems biology, is probably one of the most exciting fields in computational biology right now. There is the Institute for Systems Biology at University of Washington in Seattle, headed by Leroy Hood. At Berkeley, you have the Molecular Sciences Institute, where people like Roger Brent and Adam Arkin and electrical engineers like Shankar Shastri are involved. Stanford, Holly McAdams, Caltech, John Doyle, Harvard, Jeremy Gunawardene, and of course at the Whitehead, and now the new institute that Landa has started called the Broad Institute. There's going to be a great emphasis on systems biology. Dynamic games with learning is probably another very interesting idea that's being broached right now. The thought is the following: can we study biology as simply a study of certain special kinds of information processing systems, as multi-agent repeated game working under certain replicator dynamics? The agents could, of course, could be genes, they could be cells, they could be species or phyla. I mean, there's, and this idea is not new, of using game theory in studying biological systems, but I think what is new is probably the fact that one would actually try to study the dynamics of these games and use techniques such as machine learning techniques in this context. And Adam Arkin at Berkeley is sort of starting up this field now, and Bud Mishra at Courant and the Cold Spring Harbor is another proponent of this new development. Let me end with a suggestion that I think the time has come for a broad interdisciplinary effort and for a research center here in Bangalore that looks at in silico biology. And I think clearly we may not be able to afford to do a Broad Institute or a Biox at Stanford, but I think in fact I had a meeting with Eric Lander about a month ago, and what, in his words, we can certainly do some serious damage here in Bangalore. With that, let me stop. Thank you. Thank you.
The NP complete
right.
problem, and in the context of